Welcome back from the break. Our next presenter is Dr. Arindam Dasgupta, who's currently the Deputy Director of the Division of New Drug Study Integrity in the Office of Study Integrity and Surveillance, located in the Office of Translational Sciences in CDER. He's part of the management team in OSIS that directs and conducts inspections of in vivo and in vitro bioavailability, bioequivalence studies, and non-clinical studies submitted in support of pharmaceutical development as a part of the agency's bioresearch monitoring program. Welcome, Dr. Dasgupta.
Thank you, Ray. Welcome to the last session for today, and this is a case study. Assisting me today would be Dr. Ruben Ayala, Dr. Charles Bonapace, Dr. Sean Kassim, and Dr. Cynthia Zhang. So the case study is titled Bioanalytical Approaches to Mitigate Issues Identified During the Clinical Site Inspection. And you have already downloaded the study packet that was already available. The link was already available. So this is a disclaimer. This presentation reflects the views of the authors. It should not be construed to represent FDA's views or policies. So in introducing the case, in this following hypothetical case study, which is inspired by an FDA inspection, you will be discussing a bioequivalence study with pharmacokinetic endpoint. These studies with pharmacokinetic endpoints consist of a clinical and bioanalytical portion. Drug products with the same active ingredients or moiety are considered bioequivalent if they achieve similar drug concentration time profile in a systemic blood circulation when administered at the same dose. In this case study, same group of subjects are administered test and reference drug products in separate dosing periods. Samples of biological fluid are collected from subjects just before and at various times after dosing. At the end of this exercise, the participants would be able to evaluate a clinical study conduct of these studies, identify critical steps, and formulate mitigation strategies that may include validation experiments when issues with study conduct are identified. So what you need to do is review individual sections and answer the section questions. If you need help, please use the chat pod at the right. Wait at the end of each section for us to discuss each question before moving to the next question. So take about 15 minutes to work on the case study packet. Work on pages 1 through 6. Ask clarifying questions using chat pod at right. We will go over the section questions, which is listed on page 5 when time is up.
Sponsor.
There was another question about storage. The protocol required them to immediately centrifuge after collection. Please assume that whatever anticoagulant was necessary was in the tubes. So that was the reason why the tubes had to be inverted a few times before they
and your thinking on what is available in the case. And don't worry about other questions or concerns that are not provided. We're getting a lot of great questions, but they're outside of the case. So the recommendation is try to base your questions on what you know.
The other thing you can do is you can look at the question and try to look through the materials. That way you might be tailored towards what the question is asking you to do. So, that will, like we want to have some good discussion around this case. So please don't go past the stop sign, complete one section, then we'll go to section 1. So we will be discussing section 1 in about Thanks for having that active discussion going on during the case. So what we will do is we will clear the chat box and maybe you can put in the answers in the chat box and then we will have a quick discussion on section 1.
Arindam, this is Jeff. Feel free to go ahead and unmute yourself. We can move on to the next slide. You can advance your slide yourself. And please go ahead and lean back a little bit from your microphone. You're coming in a little bit loud. And you can go ahead.
Thank you, Jeff. So this is the question. Based on what you have learned so far in the workshop, information provided about the study protocol and study drug, if you are assigned to conduct an inspection of the study at the clinical site, what sample processing steps would you pay special attention to at the clinical site that may have potential impact on accuracy of the bioanalytical results and why? So and everybody is, it's great answers. Let's discuss question 1. So for each step from sample collection after dosing and sample processing, I would pay special attention to storage conditions, processing times, and environmental conditions of the samples and if they all met protocol requirements. Why? The drug has a short half-life and known instability in matrix. I would also pay special attention to the relevant steps, including time and storage conditions, which may have an impact on accuracy if they don't meet protocol requirements or parameters established during method validation or development. Okay, so now we will go to the next section. So take about 15 minutes to work on case study packet, work on pages 7 through 17. Again, like the section 1, ask clarifying questions using chatbot at right. We will go over the section questions on page 16 when time is up. to while you're going through the materials. That might help you. Please do type in your questions if you have questions about floppy sample collection, missing organization in note freezer temperature log is inconsistent. There were multiple ALCOA violations, missing EKGs, problems with blood collection, and some people have already come up with potential 483 items. Great. Where the time period
some of the comments are asking if there's one or multiple. So a question I would have is, what information would you be looking at to know if there would have to be more than one centrifuge in order for the times to make sense and be possible? So we can talk about that later. It's not really part of the case, but we're getting a bunch of questions about that. And so it shows that everybody is looking at the times, you know, like really carefully.
It's a good thing that a lot of people are picking up a lot of different issues, and we will soon discuss the issues. And you might see that there could be more issues than what we discuss at the end, because we try to make it from as realistic as possible. So you will probably see you could pick up more issues than we possibly discuss.
We would love to see those in the chat box.
It is the clinical question and discussion on it soon. So the chat box will be cleared. So please type in the potential 483 observations. Okay, so we are going to go into the discussion for question 2. So as most of you have picked up, the clinical site was very sloppy, and also, like you have pointed out, the sponsor probably did not do a very good job in monitoring the clinical study. So some of the questions, some of the discussions I would have in the 483, some of the 483 items could be lack of documented sampling handling times. The sample storage conditions would be an issue. Ice bath, cooling packs, ambient temperature, we don't know. There were several protocol deviations that were not reported, like late centrifugation time, late to freezer times for the samples. The freezer temperatures were not maintained per protocol. There were a lot of temperature excursions. Again, the FDA investigator had concerns with the incomplete freezer temperature records. However, this was mitigated when the firm informed the investigator that they have upgraded to an electronic monitoring system in October 29, 2013. The investigator verified that the electronic system was functional and the readings were within appropriate range. Thank you for submitting that. So we are going to go to the next section now. So please again take 15 minutes to work on the case study packet, work on pages 19 through 27, ask clarifying questions using the chat pod at right. We will go over the section question which is on page 26 in your study packet. Then time is up.
And I think we'll just go for 8 minutes this time instead of the full 15.
Okay, this is an update, so we will take 8 minutes for this section because I think most of you are now aware of the situation, so you will be able to get through the material quicker.
Supposed to be doing this in person in a big open auditorium, but really are impressed with all of the discussions on the case study chat and the engagement of people, because it's definitely weird. It's another one of the many changes to normalcy that we're all experiencing, but really appreciate your
to question 3. So I understand, so you can answer all the parts separately.
Here's one where, usually the whole point of validation and everything we've talked about today is setting up all of the different permutations that could possibly happen during a study. So now the question is, now that the study's over, we've already collected the samples, what's possible? What's possible to increase the confidence in the data?
Can you mitigate it at the clinical site? Can you mitigate it at the analytical site? Most of you are done. Maybe why don't you type in the chat box, then we can move to the discussion. So you will have 2 more minutes to work on, and then we will go into the discussion for question 3. So we are going to have a discussion on question 3. You'll be clearing out the chat box, and while you put the answers in, we will go through the discussion. So discussion on the first part of question 3. Based on the information you have reviewed from the inspection of the clinical site about the clinical study conduct, shipping manifest, method validation summary, and information about the analytical site, can you mitigate the findings at the clinical site? You cannot mitigate the findings at the clinical site. You can do it at the analytical site. The clinical site does not have any source records to address the data integrity concerns. So, what about the next question? What additional concerns did FDA investigators identify at the analytical site? The clinical site failed to transfer a substantial number of PK samples to the -20° freezer within 60 minutes of sample collection as specified in the study protocol. Additionally, the source data did not document the storage condition, example on ice or at room temperature, of the collected blood samples before they were centrifuged. The whole blood stability experiment was not conducted during method validation. Majority of whole blood samples were centrifuged 20 to 45 minutes after sample collection and not immediately as specified in the protocol. Centrifugation temperature was also not documented. Benchtop stability was validated for 6 hours 6 minutes during method validation study for the drug, but in frozen plasma and not fresh plasma. Also during validation, stability at -20 was demonstrated for only 32 days. Some subject samples were stored at -20 for at least 69 days. Additional thing to note was several samples were hemolyzed and the hemolysis experiment was not conducted during method validation. So the next part is, what potential mitigation strategies do you think FDA investigators came up with? So to assess the integrity of late-to-freezer samples, the analytical site for this study, Accurate Analytical Services, was requested to design and conduct a benchtop stability study of drug X in human whole blood up to 60 minutes at both room temperature and 4 degrees. The centrifuged plasma was to be transferred to -20 freezer after 30 minutes storage in refrigerator or room temperature. The storage condition in this experiment would mimic the sample handling procedure at the clinical site and would represent the worst case scenario for those late to freezer samples. The analytical site would also conduct validation experiments to demonstrate the impact of hemolysis on accuracy of measurement of drug X and conduct additional long-term stability experiments to extend -20 storage stability to at least 69 days. So I hope this was interesting to you guys, and let me go through the summary of the case. So validated analytical methods for quantitative evaluation of analytes in a given biological matrix are critical for successful conduct of non-clinical biopharmaceutics and clinical pharmacology studies. You have heard of all the cases which were presented today. So that was something, a feedback, that was what we wanted to give a feedback on. During validation, you are trying to mimic the conditions the subject samples would be subjected to during study conduct. Did the sample collection, handling, and storage affect the reliability of the data? Unanticipated deviations from study plan discussed in the case may need additional validation experiments to be conducted post-study to evaluate the impact on data reliability. So this is summarizing the whole case. What you need to do is, you cannot separate the clinical portion from the analytical portion. Everything goes hand in hand. So there could be situations in the clinical sites which could have a big impact on the data or reliability of the data. So each and every step through the process is important to follow and to see if the set protocols are being followed. So what you have seen here is the study has demonstrated that from dosing to analysis, each and every step has to be followed and all the required documentation has to be kept so that we can go back and reconstruct the study. So I think that's the end, and I hope everybody enjoyed this case. So I will transfer it to Sean for comments. Thank you.